Horse's Equine Innovators podcast, sponsored by Zoetis. I'm your host, Stephanie Church, Editor-in-Chief at The Horse. Everyday researchers at universities and other institutions around the world are investigating new ways to care for and understand our horses in the horse industry. In this podcast series, we talk to those innovators to learn more about their work. First today, a message from our sponsor. Core EQ Innovator from Zoetis is the first and only vaccine to protect against all 5 potentially fatal core equine diseases in a single injection: rabies, tetanus, West Nile virus, plus Eastern and Western encephalomyelitis. Talk to your veterinarian today to schedule your horse's vaccination with EQ Innovator. And now for today's conversation. Broodmare owners want nothing more than to see their new foals healthy and thriving. Sometimes foals develop diarrhea, however, which can be dangerous and even deadly. A variety of pathogens can cause this, but rotavirus is often the culprit. Our guest for this episode is Dr. Emma Adam of the University of Kentucky. Dr. Adam is a researcher at the Gluck Equine Research Center and an equine industry liaison. Early this year, she was key in helping identify the cause of some unusual rotaviral foal diarrhea cases here in Central Kentucky. Welcome, Dr. Adam. Thank you for joining us today.
Thank you very much, Stephanie. It's delightful to be here. I'm happy to talk about this. It's very exciting.
Well, thank you for joining us. We really appreciate your time. So could you describe your background, please, both as a clinician and a researcher?
Absolutely. It's probably a little bit checkered, but as you can tell from my accent, I'm originally English, went through vet school in England. Before vet school, I worked here in Central Kentucky and really fell in love with the United States. So when I finished vet school, I actually worked in racing for quite a long time in Newmarket, which is obviously Lexington's sister city in England. And realizing that I actually liked fixing horses much more than I liked training them to those extremes, I decided to embark on my sort of veterinary career proper. So I went into practice and then got my first start here in the United States at Colorado State University, and I was there for about 2.5 years. From there, I was lucky enough to go to Texas A&M, where I did an internal medicine residency, large animal. And from there, I pursued my dream, which was to become an equine surgeon. And I was fortunate enough to do that at the new Bolton Center, the University of Pennsylvania, where, as you know, we worked on Barbaro there. He was there when I was there. So I've been really lucky. I went then into private practice, both in New York, France, and then back here in Kentucky, before stopping to do a PhD, which was to really answer a lot of the questions that had developed over the course of my career as a clinician. And I'd always sort of had done a little bit of research, but really enjoyed it. So coming back to do a PhD in our musculoskeletal lab here at the Gluck Equine Research Center was just absolutely fabulous. And then that emerged into a growing need for our community here to continue to build bridges and harness the amazing amount of work and expertise that takes place here in Central Kentucky. Obviously, lots and lots of horses, lots of fabulous veterinarians and farm managers. And then with our research center as a great focus for research for horses, and as you heard from Dr. James on your last episode, the veterinary diagnostic lab being an amazing place to discover and find things, we really wanted to really focus and harness that synergy between industry and academia, and here it works really well. And so that's how come I'm in this current position facilitating those collaborations.
That's incredible. It sounds like you've had a very fulfilling career so far.
My colleagues tease me quite a lot on how many places I've worked, but it is really a joy. It's a great profession to get you around the globe.
And you've seen a lot of the globe so far, so I'm excited to hear what you do next. But I hope you stay with us for a long time.
Oh yes, Kentucky definitely feels like home. It really always has. So I'm very fortunate to be here. It is an absolute honeypot for anybody who wants to be thinking, eating, sleeping, drinking horses all the time.
Indeed. So what do you enjoy about being an equine researcher specifically?
Right. Well, we are constantly faced with problems and questions. And in equine research, we have so many different places to look for those answers. And so it is a smorgasbord of fabulous, interesting puzzles to solve, which of course, when you're interested in horses, is just a fabulous combination to try and move science forward and be part of a greater picture, a bigger team that are really pushing the frontiers forward and providing extra information and tools for horse owners and veterinarians to care for their horses better. And not just here in this country, but obviously in other countries as well.
Mm-hmm. So today we're going to be talking about rotavirus, one of the diseases that you have studied. Could you first explain what it is and how it typically behaves in a horse population?
Absolutely. So rotavirus is a very interesting group of RNA viruses. There's a large group of them, and they typically infect animals or humans in a group-dependent fashion. So we typically associate rotavirus A as being the pathogen that affects horses, and the other groups affecting other animals, domestic animals, as well as humans. In humans, it's actually a very significant disease, and the reason for the majority of diarrhea in very young babies in many developing countries. So it's a massive area of interest. And when it's a very contagious disease as well, very, very few number of particles of virus need to be ingested by a neonate to become infected. Once an animal or human is infected, they're typically, if they get over it okay, and most of the time they do, they will not be reinfected during the rest of their life. But in neonates, small numbers of particles will get into the gut. The virus needs to go inside cells, the host cells, to replicate. And the cells that the rotavirus likes to get into are the villus cells, so the small intestinal lining. The villi are like finger-like projections in the small intestine that are there to increase the surface area for digestive efficiency and also the location for many of the digestive enzymes, particularly lactase, which digests lactose, the milk sugar. And so when that virus gets in there and multiplies, the cells are destroyed at the tips of the villi, leading to huge numbers of viruses being released into the gut and passed out in diarrhea. We have a malabsorption because of the damage of that gut. We have inflammation, and we also have a maldigestion. And the combination of that malabsorption and maldigestion creates an environment that can set the gut up for other infections, such as bacteria like Clostridium perfringens, but it does primary damage in that way that causes that diarrhea to occur in these neonates, which, of course, the younger the foal, the more serious the problem.
How quickly does this normally set in?
We typically see these diseases getting hold of foals at about 24 to 48 hours if they are born to unvaccinated mares, or in the case this year where we saw a new variant, mares that didn't have immunity, or we don't think they had immunity to that new strain. So it's a problem that occurs usually in the first week of life, typically around 48 hours of age.
Okay, so how does a typical year look in Central Kentucky as far as foal illness from rotavirus? Do we see a lot of it?
So we will typically see rotavirus A occurring in foals that are born to vaccinated mares, and they'll usually get sick at around 90 to 120 days of age. And that can cause a little colic, but it's typically self-limiting. Some on-the-farm palliative care is usually what takes care of those foals. In unvaccinated mares, if we see this rotavirus A in unvaccinated mares, we will see the disease in neonates under a week of age, often, as I mentioned, a couple of days of age. So very serious and critical age. And so we usually are quite aggressive in treating the neonates with intravenous fluids. Often they'll need some pain medication. Sometimes they actually get taken off the mare so they can't nurse, because we feel that can really promote the gut's healing process. But of course, that can only be done in a very intensive care, hospitalized situation. And we usually give the neonates broad spectrum antibiotics to protect them from any bacteria crossing the gut wall and setting up a septicemia or a joint ill or something like that, which of course can be devastating. In the older foals, as I mentioned, it's usually much more palliative care, They don't often get antibiotics. They're usually sort of over it in a couple of days.
Okay. So aside from vaccinating the mares, how do broodmare owners typically prevent rotavirus?
Well, that's a great question, Stephanie, because it's a really big problem. And even though we've all really been trained very well with our biosecurity protocols for COVID-19, biosecurity protocols are really critical to preventing the disease taking hold of your farm, having an outbreak. If you have one index case where a foal gets sick with it, it is so highly contagious that even small amounts, a few hundred virus particles carried on a person's hands or a piece of equipment from one foal to another will enable that disease to cause an outbreak on your farm. And so biosecurity, using appropriate cleanliness, washing your hands, not messing with the foals too much, foot dips, cleanliness of your barn, not spreading manure on your fields, making sure that you have reduced traffic, and then, of course, disinfection and cleanliness are absolutely paramount importance. We do try to stop people using leaf blowers in barns when we have situations where we have outbreaks. That's a great way to spread disease. And we have some resources on our website, because biosecurity is not one size fits all, unfortunately, and you have to use appropriate disinfectants for this virus. Bleach in a barn situation, for example, is not effective because there's too much organic material in the barn. So you have to really read the labels and develop a plan with your veterinarian.
Indeed. Well, I look forward to sharing some of those links to materials about biosecurity in our show notes. So listeners, you can look at the landing page for this podcast on the horse or in the show notes on iTunes and follow them to learn more. So, this spring or early this year, Central Kentucky veterinarians were seeing something a little different than they typically do. Could you tell me a little bit more about that, please?
Yes, absolutely, Stephanie. So late January, February this year, we were seeing situations where farms were reporting that they would have, the first 10 foals born normally, and then each and every foal after that, for many foals, 10 or 15 foals, each foal was getting diarrhea at about 2 days of age, and some of them would colic, get a little bloated, stop nursing the mare, which of course is very serious. But the really critical factor about that is that we were seeing this go through each and every foal that was being born subsequent to the first case. We saw that on a number of farms. In fact, we believe from our phone survey, we saw it on approximately 45% of farms that we spoke to here in Central Kentucky.
Interesting.
Exactly, very interesting. And the tests that were being performed by the veterinarians in the field indicated that we weren't seeing rotavirus A in these foals, even though it sort of really fit with a clinical pattern like that. We were seeing some Clostridium perfringens and difficile on the fecal material, but the disease that is caused typically by those two bacterial organisms was not consistent with what we were seeing here, which was high volume, highly contagious diarrhea. And so with that, I spoke to Dr. Horhoff, my boss and the leader of the Gluck Equine Research Center, and said, Dr. Horhoff, I really think we have a problem here in Central Kentucky. This is something that we've got much more problem than we're normally seeing. We'll see a little bit of diarrhea, but nothing on the scale of this. And the way it was happening on farms was very, very interesting. And he immediately rolled out some of the COLA emergency response funds that we are fortunate that the thoroughbred industry have given to us for such eventualities. And so we rolled that out. And of course, it was very difficult to get reagents earlier this year with COVID testing. So we were scrambling together, and people at the Gluck were sharing reagents. My colleague, Dr. Alan Page, helped me get that stuff together. And over a weekend, we rolled out a research project to collect samples from 6 farms that we identified that would help us. Some had diarrhea, some didn't, and a number of the veterinarians in our area. So all told, we collected about 350 samples from those farms, in addition to about another 120 samples over the course of the spring from normal and diarrheic foals. It was a massive effort from our industry, our stakeholders, busy time of year, dealing with problems, a heroic effort on everybody's part. And that's one of the amazing things about Central Kentucky, is that we look for problems. If we think we have something, we really get together and sort of huddle to make sure we can identify the issues, and everybody puts in a ton of effort. I called Dr. Lee, who's our wonderful new virologist here at the Gluck. He only came a year ago. And I said, Dr. Lee, I think we have a problem, and I'm sort of concerned that we don't have the testing facilities at this point in time, because we're not seeing it on the current smorgasbord of tests that we do on these foals. And he said, oh, Emma, just bring me some samples. So, I got samples from some of the big practices around here, and some of the other veterinarians who I had been talking to and asking them to save samples. And he sent them off to get sequenced and to perform metagenomic analysis. And 12 days later, we had a very, very hot smoking gun pointing to a new variant of rotavirus that was more consistent with rotavirus group B, which we haven't really seen in horses at all, but that had 96% homology to the bovine rotavirus B. So very, very interesting. And it was such a massive hit on the sequencing score that it really is a smoking gun, even though we haven't necessarily performed all our research tests on it. So we rapidly rolled out, on the basis of that sequencing, we rolled out a PCR testing, and then we're able to do PCR testing for people, which of course is always good to be able to know what you're dealing with. And we certainly found it in sick foals, plenty. Yeah.
That's incredible. I'm amazed that you guys could be so agile, especially during a human pandemic. I guess that's testament to how well the clinicians in the clinics and you guys in the actual bench labs all work together. And I guess your laboratory is really all these different farms that you work together on.
That's right, and that is, as I said, that's one of the greatest strengths that we have here in Central Kentucky, is we all really talk to each other a lot. Central Kentucky is one of the greatest places because you can talk about horses all day long and nobody thinks you're boring. It's fantastic.
Indeed. So aside from using the new PCR test, how did discovery of this new strain adjust the activities of broodmare managers around here?
Right. Well, so even before we had the test, Stephanie, we were talking and trying to work with our stakeholder farms and anybody who we could reach to try and help them figure out what was going to be best for them on their farms. And our veterinary community were absolutely sterling in that regard. And so some of the things that helped people break the infectious cycle was to really crank up their biosecurity protocols, and that included a lot of people foaling their mares outside with minimal contact of the foal, other than obviously the IgG and physical exam check that takes place, usually on day one of their life. But if they didn't require any intervention at that time, they were really left alone for about the first week to try and minimize the possibility of transmitting the disease to them, because at that point we really knew that we had something highly contagious. So that really helped. A lot of the farms changed their practices. They reduced any kind of visitors. They changed their traffic flow of humans and vehicles in their farms. And it was a huge strain on people, to be honest, because not, as you mentioned, with COVID, there were a lot of people that would normally come and spend the season here in Kentucky. They weren't here. So the workforce was put under a great deal of strain for this. So in essence, it's all about cleanliness, the things we've learned during COVID: distance, washing your hands frequently, keeping things clean, and reducing contact. And those are the sort of the real core elements to biosecurity on any farm.
It's interesting because I passed several thoroughbred farms on the way to where I board my own horse. And I did notice this past year, during the winter, you see the lights on in the barn for the, the controlling of the mare cycles. But I saw lights outside, and I saw lights on paddocks with mares and foals. So I wonder if that's what was going on there, because that was a different thing than I had seen before at this particular farm I'm thinking about.
Absolutely. In fact, we had a presentation at our rotavirus workshop, the recordings of which are also on our website that I know you're going to put a link up to. And we have farm managers really describing how they changed the infrastructure on their farm and did indeed put up really powerful lights over paddocks that they divided and subdivided so that the mares could be outside and be observed carefully, as we do here, and foal outside with appropriate care and attention as needed. And so these lights were just the same kind of lights that you would see at the football fields. I mean, super bright. But in the light of being able to provide appropriate protection for our foal crop, that really helped a lot. So it was a very interesting talk that one of our colleagues at one of the farms gave with regard to that.
That's really interesting. I'm excited that I was seeing essentially a scientific study being carried out, or at least the recommendations from after a scientific study being carried out on the farms. That's pretty cool.
And I think some people will continue that practice next year, actually, as we go forward.
Well, it just seems like foaling outdoors is, more natural and comfortable for the mare anyway. And it's super cold, but, yeah,
That
it
is
just
one
seems.
of the big problems that we suffer with, is in areas that are either really, really hot, really that have heavy rainstorms or get really cold. We do obviously worry about the health of foals in that regard, but we have certainly, those farms are now putting in additional infrastructure to cope with those issues so that they can continue to foal outside.
That's really interesting. So what does this mean for next year's foaling season? We've got a test that we're using. We are maybe working toward a possible vaccination. What's ahead for us for this rotavirus B variant?
That is a great question, and I have to tell you, it does keep me up a little bit at night. We obviously are certainly going to be quite prepared for it, not just here, but obviously we had some anecdotal reports, although not testing confirmed, from other states within the US this spring. So we are concerned that it is still potentially out there and maybe an issue next year. And so in addition to those biosecurity protocols, as you mentioned, we are working hard to generate the data, the material to produce a vaccine. And we've been in active talks with various boutique companies to be able to produce a vaccine once we get to that point. So we will not necessarily have it ready for the pregnant mares that are pregnant now and going to foal next year, but we really hope to be able to roll that out in short order going forward, as well as potentially other tools, for example, hyperimmune plasma. But the vaccine is going to be the really key factor to move forward, so that we have a rotavirus vaccine that will cope with not just rotavirus A, but rotavirus B as well. So
Okay.
that's a big push for us.
Okay, good. So something I probably should have asked sooner, since you mentioned the impact of rotavirus on the human population around the world. Do we know if this particular variant can infect people?
That's a really good question. This particular variant we don't have any data on at the moment, Stephanie. However, we do have, anytime we have a horse or any animal, to be honest, that has diarrhea, we always consider there to be zoonotic risks, no matter what particular organism we're thinking of. So that's a note of caution for us all to think about. Now, in terms of rotavirus, we do actually classify it as zoonotic, even though we don't have any evidence per se of this particular rotavirus B infecting humans. We have certainly got some evidence that rotavirus A can do that. And actually, in the human literature about 15 years ago, all of a sudden, what popped up in the human population was a new variant of rotavirus that affects humans that actually had a segment of the equine rotavirus genome in it. So we don't know how that jumped in there, but it's now in the literature called equine-like rotavirus. So we clearly know that things can switch around and change around in much the same way as we've learned from, the wet markets at Wuhan, with the idea that that's potentially where the new coronavirus SARS variant 2 came from, that's causing our current pandemic. So I think we should be really careful about it.
Yes. And you had mentioned that it was did you say it was a bovine type variant?
Well, so we don't exactly know whether it comes from cattle in that regard. I mean, whether it jumped, but when we looked at the sequence, it is homologous to the degree of 96% to the genomes reported for bovine rotavirus B isolates. And so that is, a bit of a smoking gun that it may have come from cattle, although we have no evidence. And the farms that we talked to this year that were affected did not have any cattle on them. So it's not something just as easy as, oh, some farms had cattle and got it and some farms didn't. None of the farms that we worked with, of the 248 respondents to our survey this spring, they did not report cattle on their farms that they had or had had within the last 5 years.
It's really interesting. So, Dr. Adam, as an innovator in equine research, where do you see rotaviral research headed in the next several years?
Great question. I think we're making progress on rotavirus A because at the current time we only have a monovalent rotavirus A strain G3 vaccine, and we certainly see rotavirus A strain G14 in our 90 to 120 day old foals. So we hope to make strides with that. Dr. Lee and Dr. Wang and myself, we started an initiative last year trying to understand better how mares respond to that vaccine and how protective antibodies are developed and how long they last in foals. And so that's going to be a great bedrock to then use to study the rotavirus B that obviously we are actively writing grants for to get the funding to make sure we can continue the research on rotavirus B with the aim to get a vaccine, potentially more user-friendly diagnostic tools, for example, benchtop testing, which we have for rotavirus A that any farm, any veterinary clinic can have, doesn't need a PCR machine to do that, and to have a lot more tools at our fingertips to prevent this happening in future and to treat it better. It also has obviously some human health, One Health impacts. So we will hope to get some traction in with funding that may come from the human sphere to be able to move forward with that, because this is still a huge problem on the World Health Organization list of critical diseases.
Indeed. And One Health, that phrase, I probably didn't hear it a whole lot until maybe 10 years ago. And now, especially with, the pandemic and what you guys have experienced this year, there's just a lot more conversation about it.
Absolutely, Stephanie, and it really goes beyond infectious diseases because, as you know yourself as a rider, arthritis is a problem in horses, and they're great models for human arthritis because they have wear and tear issues. So it's a really all-encompassing approach that it means we can sort of cross-pollinate our research with what's happening in the human field, no matter what sphere it is, whether it's musculoskeletal, infectious diseases, epidemiology. And we've really got to sort of open up the toolbox that all researchers have and get all the best goodies at our fingertips for a One Health approach.
So what about the greater globe of equine research? Where do you see that going in the next several years?
Oh, that's a very exciting question. I think obviously I'm super biased and so proud of the Gluck Center because obviously the people that are here were the people that really pushed for the first equine genome to be put forward and have just produced the latest iteration, ECCAV3, which is the latest fabulous iteration of the equine genome. And so that big data approach and having these reference genomes, not just for horses, but also for bacteria, viruses, fungi, all manner of different species. I mean, that has just blown the lid off what we can do in science. It's just a quantum leap. This stuff would have been unthinkable. Even 20 years ago, it would have been super expensive. 100 years ago, completely unimaginable. So with big data, computational biology, taking an integrated approach with lots of different disciplines coming together, we are making really quantum leaps forward, and essentially we're really limited more by money than anything else. Our imagination and the tools that are coming into our labs are just taking us forward in leaps and bounds, and hopefully, we will be headed toward a much more personalized medicine approach for horses. We will also be able to have a much better comprehension of that host pathogen interaction, which is all important to understanding why some horses are going to come down with the disease and get really, really sick and some won't, with different diseases. So it's a very exciting time in science right now, I have to say.
It is. I remember reporting on the genome as it was being sequenced many years ago. And I mean, this was just sort of a figment of my imagination. I'm sure the researchers working on it saw the potential, but for me, I just was looking at the diversity of the breeds and the color characteristics and things like that, and these tools just seem to be so universally applicable. So I'm excited to see where this goes next.
Yeah, very powerful tools. Mm-hmm.
So where can listeners learn more about your work, Dr. Adam?
Well, thanks, Stephanie. That's great. We really work hard with the land grant mission here at the university, and we offer veterinary CE that is essentially open to everybody now that we're on Zoom. We're not necessarily limited by physical constraints, and you can go to our website at the Gluck to sign up for those educational opportunities. We also have a monthly and a quarterly publication. We have the Equine Disease Quarterly, which is really a splendid publication where we're looking at infectious diseases. And we have got a monthly magazine that really showcases all of the work being done in equine programs and across campus and in the Department of Veterinary Sciences, which is VDL, the Veterinary Diagnostic Lab, and the Gluck. And so every month you have anywhere between 8 and a dozen little articles on the research that we're doing here at the university that precisely relates to horses. It's all totally horse-centric, and that's called Equine Science Review. And you can sign up for that, and it just gets popped into your mailbox once a month for you to take a look at those articles. And so I think you're going to be probably sharing those links with our listeners, because our website is a place that you can go to navigate those things, but we'll give you some more direct links to get to them, so that we really like to get information out there to as many people as possible to make sure that people get good stuff to read.
Mm-hmm. Indeed, and please do look at the show notes, listeners, to see what you can find there. Lots of goodies there. Well, thank you very much, Dr. Adam, for sharing your time and expertise with us today.
Stephanie, it's been an absolute pleasure. Thank you very much indeed.
I also want to thank our sponsor, Zoetis. For more from the horse, visit thehorse.com, sign up for our newsletters, or look for Ask the Horse Live wherever you listen to your favorite podcast. If you like this podcast, please do all the things you would do to support a good podcast. Rate, subscribe, review, and share it with your friends. Please join us next time as we talk with the horse industry equine innovators.